Right, hang tight. We're going to start in a bit. Yo, Rate, Rate. Yeah. Okay, I'm just gonna say hi to the bunch that are here online. Zafi, Carmen, Hui Jen, Rayanne, Rushda, and Charlize. Hello. Hi, ladies, come. Please come have a seat here. Plenty of seats in front. Let's not squeeze at the back. Your names are? Please come to the front. Thank you. Valerie, Valerie, Valerie. Do I see your name? Yes, Valerie Yo. Is that correct? Hello, and your friend is? Xin Yu. All right. Hello. Ladies, have you all had the revision kits yet? Yeah. Great. Thank you for coming on to the front as well. Danny, Chu, Andrea, and Suhan. I think I'm gonna wait further. Okay, okay. Hello. Okay, yes. For those at home, let me just send the files on very quickly. Please help me. All right, for those online, I am putting the revision kits in the chat right now. Please make sure that you can get them. Any more surprises? No. Okay. So we're just missing an Ethan, Danny, Andrea, and a Suhan. I Okay, you have both, right? Hey, why yours is a stapler one? Sure, it works. Things are the same. No, okay. Oh, recording has started. Okay, hi people. Let's get started. Before, let me close the door. Right. Others. Now, before I begin, let me just make sure that those at home can hear and see me clearly. Come, friends at home, just give me a thumbs up in the participants panel there so that I know that you are following along. You can see and hear me clearly. Just give me a response so that I know that we are all connected well. Hey, thank you. Anaya, Zifi, Carmen, Clarissa, Ryan, Rushda, Charlize. Okay, so I'm gonna move on like that. I will keep my chat window open. So for those that are home, when you are thinking about questions and wanting to answer me, please feel free to go ahead and do that in the public chat there, so we can all learn together. And most importantly, all of you here, let's get together. So we're here for the correct subject. Let's get started. Very quickly here. So a little bit about me first, right? So I do not want us to be just listening to a person just talking and then blabbering on about the beautiful sciences of chem and bio. So a little bit of myself so that we don't feel like strangers like that, okay? I've been teaching for about 10 years and more. I studied life sciences in NUS, which was just the university's way of calling biology in a fancier way, life sciences, okay? I dabbled a little bit in psychology because I thought that it was interesting to learn about how the brain works and how the brain remembers things and makes connections to the world around us. And it helps that bio, when we learn about bio, is also learning about the world around us, right? So if you think about the topics that you have uncovered in bio, really, your digestive system, your respiratory system, so on and so forth. And then when we talk about chemistry, we will also be able to find some links between chem and bio. So that is a beautiful combination to look at together. Unlike your chem physics friends. Okay, so a little bit of myself like that in the classroom, but outside of the classroom, just like you and I have my interests as well. So just a little bit, all right. I love being outdoors generally, right? So as you can look at the left-hand side, that was a beautiful trip to South Korea last November, where we did some beautiful hikes there. It was autumn, so it was pretty cold, but very beautiful as well. I do a little bit of climbing as well in the rock walls, as you can see on the top right-hand corner. So I have the opportunity to combine my passions for teaching and climbing. So you can see me conducting a climbing course on the bottom right-hand corner. Back at the wall in NUS as well. And most recently, I just had my concert yesterday with a community band. So that was something that I am still very passionate about, making music, and it's something that I still do enjoy, very thankfully, for more than 10 years as well. Wow, when I say 10, it makes me feel so old. Okay, I hope I do not look as old as I am telling all of you that I am. But yes, so any questions, please make sure that you just get my attention. Teacher Jason, Mr. Pang, whichever you're more comfortable with, just get my attention. Or very simple and lazy, sure, will do, right? So get in touch with me, ask me questions, answer my questions as well, because we want to learn together. You're not just here listening to somebody ramble, right? If you want that, you can just go back and watch a YouTube video. Lots of them out there, right? So please learn with me. Make use of the opportunity that you can engage with me to learn together. Okay. Ray Ann, what is the issue? It is just there already there. Cannot get friends home, right? If you can just unmute yourself to ask me questions and answer my questions, that'd be much faster rather than me waiting for you to type. So, but if you can't do that, then of course, please make sure you type. But yes, hi. Absolutely. Your name is Ethan. Ethan, come. Please have a seat beside Leroy. Then, not too far back. Thank you, Ethan Lim, right? Great, thank you. Hi. Okay, so just quick introduction about the center here. These are publicly available information. So I'm just gonna like flash it in like 2 seconds. You can peruse it on your own. We are where are we? We are here, and I am at these two places, right? So our Novena outlet is our latest one, and that's where we are holding our practicals for our Sec Force next month, which is something that we can all look forward to when I do see y'all next year as well. Okay. So yeah, okay, just so practical, yay. Okay, so that's just all the things here. I really can definitely look forward to it next year. Okay, so from here we're just gonna jump straight into our content already. So okay, come, let's go. So we're gonna start with the bio segment first. Let's make sure you are here on the bio segment. Okay, Ryan is sending now. Okay, so join me in the bio one now, and we're gonna look at these. So first and foremost, we must be very aware that excretion is not in your combined bio syllabus. Hi, I would make a guess that your name is Suhan.
Yes.
Hi, Suhan. Have you collected the revision kit? No, come, let me pass it to you. Go one for each science. Alright, so just follow along with me as we learn together. Okay, so first and foremost, excretion is not in your combined bio syllabus, but I am always in the. So can you close the door for me, please? Thank you. Alright. I always want to make sure that we are kind of aware of what's going on as well. So if I were to ask you to think about one organ in the body that your pure bio friends would be learning about, what would they learn about in excretion? What organ would that be? I'm gonna try and remember some names. Rachel. Who's Rachel? Hey, Rachel. Hi, come, Rachel. What organ would they be learning the topic of excretion? Look at my wonderful drawing. Great guess. It's the kidney. Why you laugh at my drawing? Huh? Okay. It is the kidney. Okay. So that's what your pure friends will be learning lah. So we're just gonna be looking at transport humans, respiration humans. And then at this point, I totally understand that, hey, not all of us are going to be tested about for transport and respiration in our end of years because different schools go at different paces. So what I can share with you right now to adopt the perspective is, hey, we're all going to be working towards your O's next year anyway. So let's just dabble a little bit into the topic there as well. Okay? So let's get on with it together. We're going to first look at transport in humans. All right, let's go look at transport in humans. So these notes over here are actually extracted from the full set of curated notes as well. So I really may find this part of it familiar. All right, okay. So for those that will join me in learning next year onwards, you have the full set of the curated notes, which includes the chemistry and biology topics, everything in full. So this is just a quick extract for us to work on together. Just a quick reflection from what you have learned before, and then move on to the questions that follow at the back. Okay. For the red blood cells, most importantly, maybe I can bring to everyone's attention. I want to have everybody look at the idea that the red blood cell contains hemoglobin. Right? If anything, the red blood cells containing hemoglobin will be the most important feature that the red blood cell will ever have. Right? Because without hemoglobin, whether or not the red blood cell has a unique biconcave shape, whether or not the red blood cell lacks other organelles, if there is no hemoglobin, all the other features are useless. Right? So if you think about the red blood cell's primary function as to transport oxygen, hemoglobin allows the cell to bind to oxygen. All right? It needs to be able to bind to oxygen like that. Okay? Xinyu, did I say your name right? Xinyu. Okay. Can you tell me what's the importance of the word reversible over here? Hey, you cannot use the word to explain the word because you cannot find synonyms also to explain the word reversible. Wow, but reverse leh.
Connect.
Yeah, use those ideas, yes.
Carbon dioxide.
Oxygen. Okay, I'll take flip, but let's ignore the carbon dioxide you said just now, okay? The idea here is when the red blood cell arrives at the lungs, it needs to be able to bind to, hold on to that oxygen, and then when it's transported throughout the body, it gets to the muscle cells, the other organs, it needs to be able to let it go. Right? It cannot like holding on to it all the time. Right? So that reversible binding is important, and especially important when we look at the respiration in humans later on. Quick show of hands here. Have you learned respiration in humans? Hey, come on, just raise your hand. I'm not asking you to shout out loud and cheer for me. Okay, thank you. Right. Ethan, you learned respiration in humans already, right? Okay, so for this one, oxygen is going to bind reversibly to hemoglobin. Have you learned something else that also binds to hemoglobin? Forgot. Leroy then. Do you remember what I'm looking for? Sorry? What else binds to hemoglobin? Dioxide doesn't bind. That's my clue for you. Carbon monoxide, yes. Carbon monoxide does bind to hemoglobin as well. But Valerie, what's the difference then?
Is irreversible.
Wonderful. Thank you for thinking about that, right? So carbon monoxide binds irreversibly to hemoglobin. And Darvina. That sounds like a good or bad thing. Why is that a bad thing? Think properly. The words are there already. Okay, that's one step in the correct direction, right? So the hemoglobin is supposed to bind to be able to bind to oxygen. It needs to be a reversible binding. But now if a carbon monoxide has bound irreversibly, meaning permanently to the hemoglobin, Yushen, what happens next? Absolutely, thank you for saying that. Right? Okay, so which means that we have lost that one hemoglobin molecule's ability to transport oxygen. So there's overall less oxygen available in the bloodstream. Right, so we're going to be linking these two topics together. Okay, so red blood cells, the rest you can think about it on your own, right? Just a quick reminder that, hey, this thing over here is why movement of substances is so incredibly important. Okay, as I've always reminded Iris class, right? So surface area volume ratio, the topic of movement of substances, very important. Okay. All right, let's move on. White blood cells. Now, our syllabus requires us to learn two types of white blood cells. If you can see, I'm just going to jump straight into them. The phagocytes and the lymphocytes. Now, Ari, remind us, please, how do I identify these two cells based on a diagram of a blood smear? We recently worked on a question like that, isn't it? We were required to identify the white blood cells. What's the difference in their appearance? Okay, I've drawn one phagocyte and one lymphocyte. So what I was asking Ari is to do is to identify which is which. Yes, that's the correct page. How do I describe that? Do you write the words there? Or is it in the worksheet? Question 1 MCQ. Yeah. Okay. No worries. Okay. What about anyone else then? Can we name the difference in the appearance of our phagocyte and lymphocyte? Okay, so I'm just gonna pen it down like this. The phagocyte looks like this, the lymphocyte looks like this. Okay, so what are we looking for here? Suhan, make a guess. What do you think these things I'm drawing are? Exactly, thank you. So the phagocyte has what we refer to as a lobed nucleus. Can you see it's like kind of separated, right? Whereas the lymphocyte has just one humongous, irregularly shaped nucleus. I mean, the phagocyte nucleus is also quite irregular, right? But there's a certain pattern to it, the lobe, just like your detached or attached earlobes, right? So phagocyte, lobed nucleus, then the other one lymphocyte lor. Can? Alright, so not only is it irregular, it's going to be huge. A typical cell, the nucleus is large, but like it's not going to take up more than 1/3. This one you see is almost taking up 80% of the cell, right? So use that to identify the phagocyte versus lymphocyte. Okay, so next thing, the phagocyte, right? The word is phagocytosis, but in your syllabus, you cannot just throw the word down and then take it as, oh, I've described what the phagocyte does. You need to be able to say what it does as well. So take note that the phagocyte is going to engulf. It's going to take in the foreign matter. Hey, but the foreign matter is still going to be dangerous and toxic, for example. So I've got to break it down as well. So remind yourself here, right? After the phagocyte engulfs it, that is number 1. What is number 2? You're looking for digest as well. I'm going to engulf and digest so that ultimately, number 1, I isolate you from my bloodstream. That's the engulfing. And number 2, I'm going to break you down, right? I'm going to make you not harmful anymore. So that's all the function of lymphocytes. What about the function of the phagocytes? What about the lymphocytes then? The lymphocytes are the archers that are standing back on the castle wall in safety. They're not going to be down there killing the monsters themselves. They'll be standing behind, shooting arrows that will affect the monsters there, right? What are the arrows called there? They are the antibodies, right? Antibodies, right? Antibodies bind to and neutralize toxins, or even bind to the bacteria and viruses themselves. Okay, so we can think about these two functions separately. Both are defenders of the castle, so both are white blood cells. Okay, so the defenders protect. Now, the third and final cellular portion of blood are the baby platelets. Okay, so they are just kind of like not full cells, but still kind of cells. And in our syllabus, simply put, they initiate the blood clotting process. They initiate the blood clotting process. Without platelets, or if somebody is suffering from a too low of a platelet count there, they will not be able to clot blood properly. And then that's going to be an issue already, because what's the purpose of blood clotting? We are talking about something going out, we're talking about something coming in. I don't want good stuff too much of the good stuff going out, that's blood. I don't want any bad stuff coming in, right? Those are your pathogens. Pathogens, what are pathogens? Have you heard of pathogens? Livia, pathogens. Livia, not Ivory, Livia. It is actually, okay, I'll give you a clue, it is somewhere on this screen already. Bacteria and viruses, how did you know? Well done. Okay, so pathogens are bad stuff lah, yes. Okay, pathogens are bad stuff. And in chapter 7 in bio, actually, infectious diseases in humans, right? That's something we will come to learn as well. Then we will learn a little bit more about these pathogens, bacteria and viruses. What kind of harm can they cause? What kind of havoc can they wreak in our bodies, right? So pathogens are things that we just don't want in our body because they cause infectious diseases. But for now, it's enough for us to just say bacteria and viruses. Okay? Okay, so when we talk about initiating a blood clotting process, how, right? The word initiate is very important because they are simply at the start point. They are simply at the start point. They are not involved in the middle or the end. It's at the start point. How then? I have put it down into 3 very simple steps here. And if you look at this point over here, especially for number 1, enzymes, isn't it? Now, your pure bio friends will have to learn specifically about those enzymes. They will have to learn 4 different names over there. But for us, it's very simple. Release enzyme. That's all. The only new names that I hope you all have encountered in school already will be soluble fibrinogen and insoluble fibrin threads. Okay? So, let me help you understand a little bit. Instead of just, okay, just say memorize, then we memorize, right? What's the value of understanding it and writing it down as soluble fibrinogen versus insoluble fibrin threads or strands? Okay, so I'll just go to slash over here and I say threads. Okay. Now, strands versus threads. I want us to think about a ball of string, right? It's going to be very easily form a clump in your pocket if you just toss it in like that, right? I mean, if ladies with long hair, you will know, right? It's going to be tangled up very easily. And the whole idea is we want the tangling to occur if I want a clot to form. Why would I want a clot to form? So I don't lose too much blood. So that I don't want to have so much bad stuff coming in, right? So how do I ensure that the clot, the tangle can form whenever I need it, but not when I don't need it? So I have the soluble, the dissolved form in my blood first. Only when I need it, hey, convert you into the insoluble form. And that's how the clotting process works. That's the simplest way we can understand it together, right? So it's just floating around in the bloodstream, not doing anything. But the moment I need you to become a mesh, a net, I'm going to form you up like that. Okay, so that's the whole idea here. And that's why soluble versus insoluble. Okay, so hopefully this helps us understand the meaning behind the words there. And yeah, it's going to trap the red blood cells. So that's why I say the platelets are not the ones that are doing the clotting. They simply get it started, right? The clot is actually the fibrin and the red blood cells. Okay, let's go. So these are the 3 cellular components of blood. What about plasma then, the non-cellular portion? Now, simply put, plasma, can I ask you all to think about it as just a rich soup? Plasma is just soup. Lots of nutrients, lots of glucose, amino acids, vitamins, your proteins, ions. Just that this time the soup got some a little bit of a dirty stuff as well. So you definitely have your carbon dioxide. Okay. So this is a soup that you will not want to drink directly, right? But it is a soup in the sense that, hey, if you have like a chicken soup, the meat, you have your red blood cells, the carrots, you have your white blood cells, your potatoes are your platelets. Then the soup itself, how is plasma then? Okay, so it contains all the other dissolved substances, good and bad. So the good stuff here, I usually don't urge us to memorize it just like that, right? Because the more we learn the future topics, it becomes more familiar. So no need to memorize them all at once. Maybe only, please make sure we are able to name nutrients like glucose and amino acids, as well as some hormones like this. Okay, so since I'm highlighting insulin over here, Sohan, can you tell me what insulin does? Was the last word you said clot? No, nothing to do with clotting. Ethan, function of what? Hey, hello, follow. What's function of insulin? Hmm? Say. Say lah, you wrong, then we learn. You correct, then we continue. Say, cannot, you must say a word. What does insulin do? Must dare to make mistakes, ah? You say if you're wrong, then we learn. If it's correct, move on. Come. Function of insulin. Ethan. Try. There's something in your head one. Tell me. Come on, Ethan. Louder. Whisper to him louder. He is telling you, listen to him. Ethan. Yes, thank you. Okay, must be able to remember. So today must remember your friend Leroy told you insulin helps to lower blood glucose. Okay, Ethan? And it will do you good to write that down. Okay? Thank you. Well done, Leroy. Right, so insulin helps to lower blood glucose, right? It helps to lower blood glucose. And the word helps to is very important because insulin doesn't do the lowering itself. Please remember that. Insulin doesn't do the lowering. Insulin goes to the cells and says, can you do this and this? Which eventually results in a lowering of blood glucose. Right? Irie, what does insulin do? Hey, that's why he said already, yes. How? Can you tell me what cells then insulin targets in the body? Rest of you better be thinking, Alright, something that we always have in class here is even if I'm not calling your name, you better be thinking because I could call your name next. Alright. What are the target cells of insulin? What organ looks like this? The liver, thank you. Irie, come on. Since when the liver is a what? The liver is a triangle this way in the right side of your body. You find your lowest rib on your right side, your liver is here. It's a triangle. It is the largest internal organ. It is in that rough. I mean, it's not going to be right angled, but Pythagoras theorem triangle lah. But it's roughly that shape. Understand? Liver and one more. You know what's the other organ? What other? Muscles. Thank you. Insulin targets liver and muscle cells. Okay, insulin targets liver and muscle cells. What else? Yu Xuan. What does insulin do then? What does insulin tell those cells? How? Yeah. We learned that in nutrition in humans. Digestive system.
Breakdown excess glucose.
Not breakdown. Convert. The word is excess, correct? So converts excess glucose into must telepathy a bit. Wonderful! Glycogen! Thanks for remembering that, okay? So it converts excess glucose into glycogen to be stored. right? Livia, what else then? What else does insulin do? Other than to stimulate the cells to convert excess glucose into glycogen for storage. Why would there be so much glucose in the cells?
Because the blood glucose concentration.
Okay, so blood glucose concentration is too high. That's the whole point of insulin being released. So actually, that would have been the second function. The first function that we need to state is actually insulin tells these cells, hey, absorb more blood glucose. There's too much glucose in the blood. Can you take some in? Then blood glucose will start to form. All right. So the first function is to increase the amount of blood glucose absorbed by those cells. Okay, so please make sure we can link all these things together. The more information we can link together, the less small things that we have to memorize, isn't it? You just remember for one point, everything flows. Okay, right, link it all together. But coming back over here to this topic here, right? So like I said, all the substances transported within plasma going to be linked like that. Carbon dioxide and oxygen is going to be linked via respiration. Let's very quickly talk about urea then. Valerie, what can you tell us about urea?
It stops your wasting.
Only one word is correct in that phrase. Thank you for saying waste. Urea is a waste product then, right? Urea is produced via the not destruction. Kind of sounds like that. Rachel, let's hear from you. The wonderful elimination of, and I love that you remember the word excess. Thank you. Well done, elimination of excess amino acids. And from this point, I hope that dear Ari can remember again. Ari, where does this happen at? Yes, thank you. Well done. This happens in the liver as well. Okay. So when excess amino acids are deaminated, they're broken down, and this happens in the liver, what do you form? Urea. Okay. So what happens to urea? Does it just stay floating in the bloodstream? Urea is a waste product, right? Why would I want the waste product to be floating in my bloodstream? I'm going to remove it. How is urea removed from the body? It's the thing that most people confuse it with. What does it sound like? You no, you shouldn't. Yes, you should. No, you shouldn't. No, you shouldn't. Xinyu. How does the body remove urea? What's the word? Nope. That's not really the correct word. Urine. Thank you. Okay. So the body removes urea through urine. Can? Right? Urine contains urea. It is not the same as is. Okay. And that's actually the extent of what we need to know about the function of the kidneys. Right? Remember, just now I said that topic of excretion, your pure bio friends will learn the whole thing. But for us, we don't need to go down that whole rabbit hole. It's just removed in urine. That's all. Okay? Let's move on. We do not need to learn about the blood group systems. That's for your pure bio friends. So you don't have to cross out the whole thing lah. I'm just putting it here so it's easier for us to see. But if not, you can just, it is there for you to read up on your own if you want. Okay? Let's go. Blood vessels though. This one very simple. Let me just stress again. A for arteries, A for away. A for arteries, A for away. And it's always with respect to the heart. So if A for arteries, what can I help you with? The veins. Veins bring blood in to the heart. Right? So you have the word in there as well in the word vein name. Okay? And the primary word we want to remember about capillaries is exchange. Right? So simply put, arteries and veins are just for transport. I bring you there, I bring you back. But any exchange that occurs, where are the bus stops? Capillary. Right? Only at the bus stops can you board and alight the bus. So on the road, there's the arteries and veins. Bus stop is capillary. Okay? Think about that. For the structures here, please remember the most common comparisons are between the arteries and the veins, right? The most common comparisons are arteries and veins. Primarily, wall thickness, lumen diameter. Suhan, have you heard the word lumen before? Ken, I'll take it. Thank you. Right, the lumen refers to the space inside the vessel. Ken, right? So if this is a blood vessel, right? For those at home, just have a look at my video here. If this is a blood vessel, this space here is the lumen. Okay, right? So arteries typically narrow lumen. Veins, large lumen. Okay, that's it. Very simple understanding. Okay, so why then? Why do arteries have such narrow lumens? Irene. Come on, come on. Narrow is small. Artery do what, Irene? What did we just highlight above? Away from the heart means go to where? So I want to transport so little only. Thank you. Irene, come on, we gotta remember things, Alright, the arteries have very narrow lumen. But Livia is still missing one word actually. It's something about high blood pressure. Wow, you're giving me something else already. So if you talk about why do arteries have narrow lumen, it is got to do with the high blood pressure. Just one word. Yeah, flipping it around. Withstand is for thick walls. Thicker the wall, when the pressure is very high, the vessel won't burst lah. Fair? Can? Alright? So this is to withstand. The high blood pressure. Then narrow how? Narrow is something high blood pressure. You remember. Well, that means in a 2-mark question, you will get 1 mark. You can only tell me the thick wall is to withstand. Then forget the second point already. So come, let's all work together. Get that second point in the bank. Narrow lumen. What about the high blood pressure? What else can a blood vessel do to the high blood pressure? What is pressure? Can we think of if the blood pressure is very high, the blood flowing very fast or slow? Fast, thank you. Right? So as it leaves the heart, the blood flowing very quickly. By the time it gets to the cells at the other end of the body, would we want that blood to now suddenly flow very slowly already? So then what? What's the word there? What's the word there? Start here very fast, very high pressure. Reach the cell, also very fast, very high pressure. I want it to remain constant. One word for that is simply to what's the word? Maintain. Thank you, Olivia. Maintain the high blood pressure. Okay, very important. So the rest of the world will be getting that 1 mark in this 2-mark question. I hope that after today, all of you here will be those that are getting the 2 marks there. Please remember, right? Thick wall, withstand, tahan that high blood pressure so it doesn't burst. The lumen diameter in arteries are narrow so that the blood pressure remains high throughout. Right, consistently high throughout, so I can deliver the delicious oxygen and glucose quickly to all the body cells there. Okay, well done. All right, So typically it's just these two. For the exchange function of the capillaries like that, we can look at the right hand side, right? The wall itself only one cell thick. One cell thick only. That's the thinnest you can get. Okay? Whereas these two, the arteries and veins, multiple cells because it's layers of muscles. Okay? Right? Then for the lumen though, I put down the words very narrow over here, but I want you to optimize it. Instead of very narrow, we're gonna write down something over here that I want you to. And I'm gonna draw for you, and you're gonna tell me what words I'm looking for. Okay? This is a capillary. So I have drawn for you the one cell thick wall, isn't it? So this cell, this is the wall, one cell thick, okay? Function of the capillaries, we typically actually think about the red blood cell, okay? So red blood cell flows through. Let me show you how the red blood cell flows through here. So how wide is the capillary lumen here? One cell wide. Specify it. Can? Thank you. Right? So instead of very narrow, we say one cell wide. Like how narrow is it? It is one cell wide only. So this means think about yourself. After your assembly, you all go back to the classroom. The whole hall so wide, the doorway, but by the time you get to the corridor, it gets a little narrower, right? Then by the time you get to your classroom, imagine only one door open. You all can only enter one at a time. You all will slow down, isn't it? Do we want this to happen in the capillaries? We do. We do. We want red blood cells to flow slower in the capillaries. Why? Thank you. It's the word exchange there, isn't it? Think about the bus stop example again. I'm very glad that buses slow down enough to a complete stop before I can board and alight. If you just continue, bus the bus stop, you're not going to be able to board and alight, right? Right? But of course, in the bloodstream, it cannot stop entirely. So it slows down as much as possible so that the nutrients can go to the cells, the waste can come into the blood. And that's to facilitate the exchange that you mentioned. Okay? Right? So blood cells have to slow down. And that's actually why we have a loss of pressure only in the capillaries, not in the arteries. Okay, so that's it. Usually just these two only. The third one, if I were to bring up one thing, would be valves are present in the veins only. Okay, right? So if you think about this, we have talked about arteries having their high blood pressure, venous pressure. The blood pressure in the veins are going to be very, very low. And that means that, hey, the blood may not always flow in the correct direction because the pressure is not there to push the blood forwards. Okay, so what's going to happen? Valves are like one-way doors. You can only ever push the door open. So if you want to go the other direction, how? And now I remove the ability of your arms to do any pulling. Just gonna be bumping into the wall. You cannot push that one-way door, and then you'll be stuck. But this ensures that you can only flow in one direction, okay? Let's think of yourself as that bumping into the wall thing. All right, that's it for blood cells and blood vessels. As a quick recollection, for the structures of the human heart, it is something that we do have to be able to label and identify everything, but this comes bit by bit, right? So I really remember that how we talked about this structure of the heart there. Wonderful, right? So maybe you can share that thing with Livia next time as well. All right, but for the rest of us, please make sure I need y'all to look at this structure and go through it one step at a time. Don't just look at the whole thing. Oh my goodness, there's so much here. And I do hear you. It's definitely going to be overwhelming if I just tell you straight up, okay, memorize everything. It's going to be difficult. Okay? Right? What we can break this down into is to draw a heart. Right? Like that first. Right? Very simple. If you draw a heart like that, everybody knows how to draw this heart. I can't draw this heart. I can't draw this heart. I can draw this heart. But what am I going to do here? I'm going to quarter it. Those are my four chambers, isn't it? All right? I'm going to look at this right now. Hey, what letter does this form? It's a V. So therefore, these are the ventricles. Isn't it? What letter does the alphabet start with? Look at that. I have my four chambers of the heart. Okay, and when I'm looking at the structures of a heart, we're looking at it in a mirror image. So therefore, look at your right hand. Then you write down left. It's just gonna be opposite. Look at your left hand. Then this will be the right side of the heart. That's it. Okay, so use this to remind yourself, hey, these are the chambers of the heart. Now, coming back to the letters then, the alphabet starts with the letter A, and that is also how blood first starts its journey in the heart. So blood always enters the atria this way. It's going to trickle down to the corresponding ventricle. It's going to exit the heart from the ventricle. So tell yourself A to V. And tell yourself A to V, just like how the alphabet flows. And use it to help yourself. Like that only. Okay? Dervina. Why did I use different colors for my arrows here? Blood. Thank you for saying that word there, blood, right? So yes, it represents the oxy and deoxy blood flowing through the heart. That's very simple, right? So maybe some of you in school may have been introduced to this as well. You will know what Lord is. Lord. Wonderful chorus there, right? So, nope, wrong colors. Left oxy, right deoxy. Blood in the left side of the heart is oxygenated. Blood in the right side of the heart is deoxygenated. You could use that to help yourself remember as well. Okay? Okay? And with regards to blood vessels, the naming, blood vessels always have two parts to their name. I am Jason Pang. The blood vessels, first name is what organ are you connected to? Second name is what direction is the blood flowing? In terms of direction, we look at the top of the page as well. We always come back to this. A for artery, A for vein brings blood in to the heart. It's always with respect to the heart. So very simple. All right, what is the word we use to connect to the liver? The word is hepatic first. Okay? So when you hear the word hepatic, it's going to be connected to the liver somehow. Right? So then first part of the name done. Hepatic something. If it's hepatic, look at this, artery. If it's hepatic artery, if I draw a line like this and I tell you this is the hepatic artery. How do I spell hepatic? Okay. Now what's here? What's here? Just tell yourself artery. A for away from the heart. So therefore, let me change. Circle and square. Which one is the heart? Circle away from the heart, right? Then this one is hepatic artery. And the other side is what? Liver. Well done. Look at that. So blood vessel names is just like that. First name, last name. First name, what organ? Second name, direction with respect to the heart. Okay? We only need to learn 4 words for organ association. Hepatic is liver. Sorry? Pulmonary. So MN. Thank you, liver. Pulmonary is lungs. Can? Jingy, can you tell me something else? You have hepatic for liver, pulmonary for lungs. Where did they come out from? That is the 2 words, yes. I think I have lost you in the last 5 minutes. What I'm talking about now here is blood vessels got 2 parts in their name one. First part is what organ they are connected to. Second part is, are you artery or vein? That's all. So pulmonary is associated to lungs. Hepatic associated to liver. We have 2 other words that associate to organs one. Do you know what they are? Hepatic is still liver. Renal is not what I'm looking for. Right? Sorry? You tell me lah. No, you just give me the word first. No, that's the point. You must figure it out. So what? Renal. Thank you. Renal what organ? Kidney. Thank you. So that's the third. What is the fourth and final one? Yeah? Valerie? Rachel. Oh, we don't learn intestinal. No, also. Oh, I think so, huh? No, it's not. It branches out from the aorta though. All the arteries branches out from the aorta. Okay, so on the next page. Second half. Don't flip so much. Yeah, there. Coronary. What organ does coronary tell me? What does coronary tell me? Heart lah. Like, coronary artery, heart to heart. Doesn't make sense, isn't it? So let's take this quick look. Go and see this page here. What should we highlight here for coronary arteries? Oh no, not highlight. Let's write it down then. Okay, coronary arteries. What do the coronary arteries transport blood to and from? It is from this one. There's a slightly more specific way to describe the transport. It is from aorta to heart muscle. So it's still heart to heart, isn't it? Right? Just that from the LV, the left ventricle, it exits through the aorta, looping back onto the heart. But this time, from the outside. Can you see this one? I see my cable here. This is the aorta from inside the heart. It's gonna come out to the outside of the heart here. So blood from inside the heart, right? The cells cannot use because the blood is going in and out too quickly, right? Every time you contract too quickly, right? So then the heart made of cells itself, right? It needs to be alive, right? So it needs oxygen and glucose, but it cannot take from the inside because blood is going in and out too quickly. So therefore, when it branches out, this one go to the liver lah, kidney lah, brain lah. But it also has this one tiny loop that goes back to the outside of the heart itself. Then from here, it will give the heart cells the oxygen and glucose. This is called the coronary artery. Is that clearer, Valerie? Thank you for giving me that. So that I can explain to everyone just now. Okay, so that's the coronary. So again, forwards Sing you want forwards.
Pulmonary and coronary.
Thank you. Okay, please remember them. Right, so that's kind of it. And then we have the 4 doorways, the valves, they are listed there as well. On the left-hand side, we have the ones on the right of the heart. Left-hand side of the diagram, right-hand side of the heart. Can you see that? Okay, right? So pulmonary and tricuspid valve on the right side of the heart, whereas the bicuspid and the aortic on the left side of the heart. Okay, Ari, what does bicuspid and tricuspid refer to? Do what? Okay, I'll take sections then. Right, so for example, if you look at the door right here that we just entered our classroom from, if I were to refer to that as a valve, that would be a monocuspid valve then. So it's door with just one, it's doorway with one door. So a bicuspid is 2 doors. This way. Can? Right, a tricuspid got 3. That's it. Can? Fairly straightforward. So just for you to identify it more easily, bicuspid is on the left side, so it's 2 doors only. Now for the aorta, the aortic and the pulmonary valve, it is simply connected to the vessel. LV contracts to pump blood into where? LV contracts to pump blood into the LV is left ventricle. So when the left ventricle contracts, blood goes into the aorta. So therefore, the doorway is called the aortic valve. Okay? Look at your notes now. Find the RV. When the RV contracts, where does blood go to? When the RV contracts, blood needs to go into the pulmonary artery. So therefore, the valve there is called the pulmonary valve. Okay? When RV contracts, why does blood go into pulmonary artery? Why does it not go back up into the right atrium? Excellent. Think about one-way doors, isn't it? Right? Think about one-way doors. And the thing about the flaps of a valve, they always point in the correct direction of flow. From RA, oh, where's my laser? From RA to RV, direction is this way. Look at the flaps. From RV, pulmonary artery, this way. Look at the flaps. Okay? And it's not just a diagram thing. They actually kind of do look like that as well. Right? Obviously not as obvious. It kind of does have that look as well. Okay? And if you ever forget what it does, yeah, use your hands. Form a heart shape first and then open it up. Okay? So therefore, my fingers are the flaps. Look at that. Blood, when it flows, it will be able to push the hands down. Blood can flow down. But when blood wants to come up, what's it going to do to the flap? It's going to push the flaps, touching each other, and now I've closed my valve. Okay? Okay? So that's how I can help you understand how valves function. And with that, right, please make sure all these tiny little things here, top it up with what you have learned in school, in your notes. Let's move on. For the graph here, please make sure we are very aware of how when the valves open and close. And for the entire graph over here, please just focus on the intersections. The intersections are where most of the questions will ask about. Okay, so the intersections are what we're talking about here. Now, AV, atrium, and ventricle. So there are two AV valves, your bicuspid, tricuspid. Then your semilunar are your pulmonary and aortic valves. Okay, typically we don't have to spend too much time on this, especially for combined bio. So I'm just gonna leave it over here. If you encounter any questions next time, please just make sure to look through our notes in further detail. But for the last part of this topic over here, as we mentioned, coronary arteries. Now, why is there so much more emphasis placed on the coronary arteries? Because, hey, look at that. Then the heart muscles, how? They cannot receive enough oxygenated blood. What's the process that I need us all to think about? The process here is, if not enough oxygen, why will the cells eventually die? What process will the cells not be able to undergo enough of? Thank you, Irie. I love that. Thank you. Respiration. Right? Not enough oxygen. As well as glucose, isn't it? So if the cells cannot receive enough oxygen and glucose, the cells cannot undergo enough respiration, cannot release enough what? Energy. Thank you. And then die. Okay? Never go straight to the cells will die. You can only go to die if your prior point is energy. You must go to energy first. Then you say eventually die. It will never be immediate death. So always bring your story all the way to energy. Then you can ever say causing it to eventually die. Okay? Alright, wonderful. Thank you for remembering that. So why will it narrow? Why will it become blocked? Right? We need to think of fats and cholesterol in the diet. Right? Moderate consumption is actually required. We do need some fat. We do need some cholesterol in our diet for healthy growth of tissues and cells in our body. However, too much of it, then the bloodstream, how? Fat and cholesterol builds up inside the vessel. So this is the blood vessel. This is the amount of space that blood has to flow, isn't it? Normally. But now, if the fat and cholesterol builds up like that, now only got this amount of space to flow. Do you see that? Right? So there's less space for blood to flow. Will the cell say, okay, lor, I just received less blood, lor? No, right? The heart, in its ever sacrificial and human loving form, will therefore, okay, I got you. Let me pump harder and faster for you. But if this prolongs, the heart is just a muscle. It will tire out. And that's where damage occurs. Okay? Alright? So very common question type is linked to heart attacks. Like, why will the blockage result in heart attacks? Talks about oxygen, glucose, respiration, energy, eventually die. Okay, let's go. Next topic, respiration. For respiration, fairly straightforward at the start because we would have learned most of it since primary school even, right? So the structure to the nose, the structure of the throat, okay? So fairly straightforward, I'm not going to spend too much time on this, except when we come to the trachea, we need to focus on mucus. We need to focus on cilia. Alright, within the trachea and within the trachea, so the airway, we have mucus and cilia. Now, a very common error is over here. Cilia. What's the function of cilia is to sweep. But students often misunderstand this to say, oh, the sweeping action of cilia, what does it do? It sweeps away dust. Incorrect. Why? I tell you later, you go home, take your broom or your duster, flip it out into the air. Are you going to be able to sweep any dust away from your house? You'll put a dog big pig, you. You're not gonna be able to sweep the dust out from your air, isn't it? So the sweeping action, do not link it to the sweeping of dust away. What can you sweep up instead? You can sweep up the sticky solids in the trachea. Where did that sticky solid come from? Or even liquid, right? Now they bring in mucus. So the first step to describe function in trachea is mucus first. Mucus traps dust and pathogens. Then later the cilia sweep away. Can? So trap first, then sweep away. Where am I sweeping it towards? Away from the lungs, yes. Where am I sweeping it towards? This is something that we must be very clear about, is to sweep to the throat. And yes, that face of disgust forms on Yushen's expression. To be swallowed or expelled. Right this very moment. I hope none of you are sick, isn't it? None of us, including me, are sick. So we are actually constantly swallowing the delicious mucus that your trachea has absorbed, has trapped all those dust away. Yushen, why are we falling sick now? If the mucus all this dirty thing, right? Then we're swallowing it, leh. Then how? What happens to it? Where does it go to? If you swallow something, where does it go to? Give me the name of that part. Something, then stomach first. What's that something first? The esophagus. Very good. So when it gets to the stomach, that's where I want you to tell me. The dirty things go to the stomach. What happened to it? And nope, not digested. It'll be broken down, but not digestion. All the bad stuff will be killed by what? Stomach. What's the acid? Hydrochloric acid. Can? Okay, so anything that you all the dust and dirt there, you swallow it down. What? It goes to the stomach, hydrochloric acid is there to protect us. Can? Right? Only when there's an excess of that mucus, and that's where we fall sick. Then you have excess mucus, and you have to, you know what happens next. Okay, then that's where it has to be expelled. All right, so please keep in mind, we do not sweep dust away. We sweep the mucus that has trapped the dust. Okay, let's move on. Gaseous exchange, easy, because why? It's not learned since primary school, Gaseous exchange. Last time, you referred to the alveoli as the air sacs, no longer in secondary school. Okay, so the alveoli is where gaseous exchange occurs. Please keep in mind, oxygen goes straight into red blood cell. Because red blood cells have what? Hemoglobin, thank you. Right, carbon dioxide is from plasma into the alveoli. I want to bring to our attention this thing over here. If you have a colored marker, you can do that with me over here. What does it represent? What does this represent? After you've done this highlight, look at the table that follows at the bottom. What does this represent? Thin film of moisture. Thank you, Olivia. Right, we have it over here. Oops. So what is that for? We have dissolve, then diffuse. Right? Oxygen is in the gaseous state, isn't it? We do not want gases in the gaseous state in our blood. There are no gases in the gaseous state in our blood. Right? They're all dissolved. So we let it dissolve first, Right? So rather than you dissolve as a gas first, Well, means you must enter the cell as a gas. No, 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 no. You dissolve first. Then equals diffusion. So much simpler. Okay? Right, so dissolve and diffuse. This is something that is commonly overlooked, right? But it's very important. But other than that, if you look at this, look at that surface area to volume ratio. Steep concentration gradient. Decreased diffusion distance, right? Surfaces again. What topic comes to mind? Movement of substances, right? Your diffusion and osmosis stuff. So once more, if you've now realized, well, actually, my movement of substances is quite weak, please go ahead and go and look through that again. Because even though EOY never state, right, can it still come out? Absolutely. Movement of substances is one of those topics that are found throughout. Okay? Right, so go back and review that. Let's move on. Breathing and respiration, please take note that they are different things. We can only control one of them. Leroy, which one can we control? Breathing or respiration? Breathing, because you just hold your breath. That's breathing. But you cannot, no matter how hard you concentrate, stop respiring, stop respiring. If that happens, you die. Okay? So thankfully, we do not have the ability to stop or control our rate of respiration directly. I mean, we can cause it to go up. Let's go and exercise now. It will go up already, ma. Right? Okay? Right, so please keep that in mind. There are two entirely different processes. Breathing is simply pushing air in and out of our lungs. That's a breath. Right? What is respiration itself? Respiration is a chemical process which releases energy. Okay? Right? As Ari and Yusha have reminded us, releases energy. So, aerobic versus anaerobic. If you look at my screen over here, it's nicely colored, right? Aerobic, anaerobic. The biggest difference that we must bring across first is this. And aerobic involves oxygen, anaerobic doesn't. Second comparison is lactic acid. Lactic acid is toxic at high concentrations. And lastly, what would another difference be between the two types of respiration? That's the first one, oxygen, right? Need oxygen, don't need oxygen. Second one is produces something toxic. Those two relatively harmless. The last comparison is? So carbon dioxide and water is compared to lactic acid. This one is toxic. This one is not. It's relatively harmless. Thank you, right? Energy, right? Valerie, what about the energy?
One.
Which one? Tell me. Very good, right? Aerobic respiration releases lots of energy. Anaerobic, so much less, right? You do not need to know about the comparison of the numbers. You will learn that in JC Bio next time. Should you go down that path? But the number is like a 1:16 ratio. Like a 1:16 ratio. It's a lot more. Okay? Right? What is 1:16%? A lot. Okay. Just a lot more, right? So our syllabus is just huge amounts. Very little. Okay? Right? So when does anaerobic respiration occur? You release so little thing, you release so little energy, you release toxic substance. Why would I ever want to engage in anaerobic respiration? Got limit. What got limit? For what process? Yes, right. There's an upper limit for aerobic respiration. And that's what we train to do. That's what athletes do, right? Athletes, they train, they increase their aerobic capacity. So then they engage in anaerobic later. But for normal folks like us, after a while, we're very easily tired, right? And then anaerobic kicks in, and then so we start getting more tired. Right? So that's what, if you're wondering like why do people train, they're pushing their aerobic capacity. Right? So they engage in anaerobic later, which means they continue that humongous amount of energy production for longer, and they delay the production of a toxic substance as well. Okay, so the limit, we focus on actually oxygen again lah. As long as the cells can constantly obtain enough oxygen, they will be able to undergo aerobic. Not enough oxygen already? Okay lah, I will continue whatever aerobic I can, top it up with anaerobic. Okay, so that is that. All right, come, let's go. So what the lactic acid thing over here? Something I have realized is that students like to just oxidant debt. Just throw the word out there, oxidant debt. And yes, you are hearing me correctly. It is spelled with a B but pronounced without. It is an oxidant debt. Right, so what is the oxidant debt there? Actually, the words here don't need you don't have to write down the word oxidant debt. What's more important is to explain what it means. Okay. And Irie, where does this happen at? What organ breaks down the lactic acid? Yes, the beautiful triangle. The liver. Do you see that, Ari? Okay, so it happens in the liver as well. So again, linking the functions of the liver across many topics. Okay, all right. So go back and read through this on your own. Let's move on to the last part over here, harmful effects of tobacco smoke. Our syllabus only requires us to learn these three harmful components. Okay, so we have nicotine, tar, and carbon monoxide. And carbon monoxide is what we were talking about just now, isn't it? Carbon monoxide. Okay, all right, so these three, I think is fairly straightforward. It is a small component in our syllabus, but it's very important. Okay, so please make sure you read this up. Right, for nicotine, if I were to just highlight a couple of things here, nicotine, usually we're talking about addiction, as well as a spike in heart rate and blood pressure. That's for nicotine. For tar, we are talking about carcinogenic. Any guesses for what carcinogenic means? Cancerous. I love that. Causes cancer. Increases the chances of developing cancer. It's just something about cancer. Okay? Thank you, Rachel. And then what else is it going to do? Paralyze cilia. So just now, the cilia sweeping action. What does it sweep again? Mucus, isn't it? It doesn't sweep dust, mucus. But now with the tar, how it just stuck there. Okay? Alright? So therefore, because I am stuck, I cannot push the dust away. I cannot push I cannot push the mucus away. But then tar is also there, isn't it? So bad thing plus bad thing. Then the other cells there, still got bad thing. Let me produce more mucus. Then more and more things. What happens? It irritates the lumen, the airway, isn't it? Then how? Your body will want to remove it. That's where. What will you do? If your airway stuck, blow out the mucus. If it's deeper within, you start to cough. Yes, we start to cough, isn't it? And coughing, when done gently, has no harm to our trachea. However, we hear the Uncle Ape at the coffee shop, that one, here, very eroded. Right? It causes harm. It wears away the lining of the airways. Alright? And this may even cause further harm right into the lungs. What are those tiny little things in the lungs called? What are those things in the lungs called? Tiny little balloons there. The alveoli, they may start bursting. Because what is a cough? It is you take in air, suddenly out very quickly. Then you in very quickly also, then burst out. Okay? Alright, so that's a thing that we think about as well. What else does tar do? This is an actual image, right? So a smoker's lung is black and scarred with tar. Right? And what does carbon dioxide do there? Look at that. Alright, what Yushen told us about just now already. Okay, binds irreversibly or permanently with hemoglobin. So less oxygen present. Alright, let's talk about, of course, go ahead. No problem, thanks for asking. Alright, so we talk about the plug here. What is the plug mostly made of? What's plug made of? Two things that we highlighted earlier on already. And thank you, fats and cholesterol. Okay, so this is the image I want you to have in your head. Okay? And that's it for these two topics. Okay, so 5 very simple MCQs here for the Okay, come, let's work on it together. Let's work on it together. For question 1, I hope you find this familiar. You. Alright, flip all the way. Right, we're skipping exclusion, like I said already. Familiar or not? No meh? Ah, okay. Okay, come. Can find the worksheet. Just flip until you continue to see it. Okay, so here we have blood smear. Here we're looking at phagocytosis. What cells are we thinking of then? The white blood cell, right? Obviously, which white blood cell then? Phagocyte law, correct or not? Phagocyte. What are the two words we think of for the function of phagocytes? Engulf and ingest is it. Either one. To engulf is to ingest, and then we digest, we break it down. Okay? So based on this blood smear, how do we identify the lobed nucleus? Thank you. And then we have it. Correct or not? Lobed nucleus. Let's see. We're done. What's A? Quickly. Easy. What's A? Red blood cell, exactly. Thank you. We notice that by the brighter middle, right? Because what is the shape of red blood cell? Biconcave. This means the center thinner, right? What microscope are you using? Light, also more like a far field center. That's why it's brighter. Use that to identify your cells. Okay? Is that the first time you're hearing this? Okay, so okay, then good. Right? At least I have the opportunity to share this with you. Use this idea of biconcave, link it to center thinner, more like pass through. Use this to identify your blood cell. Okay? Awesome. Right. And then what is D? What's D first? Is that soup? Nothing there, so it's soup. Clear? Right. This is similar to cytoplasm in a cell. When the label points to nothing, you are pointing to the cytoplasm in the cell. So in blood, you point to nothing, you point to plasma. Alright, last but not least, B. Very good, Irene. The lymphocyte then, right? The humongous nucleus, right? Let's go. Okay, so this is something that I haven't talked about in the notes portion, so that I can look at it over here. We are looking for valves open, valves closed. Let me highlight close first. If the valve... Wait, did I ask you a question? Wow, what's different? Oh my goodness. Oh! Hey. Yeah. No. Hello. No. Is there a difference? Then flip Continue flipping until you see this. Continue flipping. Are you on the correct one? No, the correct one. Yes, Ishan is correct. Yes. Have or not? Yes. Yes. Okay, you're the correct one. Alright. Yes, of course. Go ahead. Okay. Alright, friends at home, make sure you are flipping to the correct question set. You must have the bracket one. So just now y'all were looking at the one for the pure bio people. They will need to know about blood typing, which is one just now we crossed out, isn't it? The question one y'all saw was the blood typing one. Okay? I never closed my door again. Okay, valve closed. Tell me first. The valve is closed. Is blood trying to flow in the correct or wrong direction? Wrong. Can you close the door for me, please? Thank you. Thank you. You are correct. So tell yourself, hey, if a valve is closed, this means that
Please.
it's trying to flow in the wrong direction. Of course, go ahead. It's trying to flow in the wrong direction. I mean, I put trying to because it's not like a real living thing that has choice. Okay? the next thing is we identify the valves here. You know what, Ivy, come. I want you to figure this question out for us. Remember what we went through previously in our drawing there? We talked about the valve thingy. So now you choose one of the valves. The bicuspid. Bicuspid is between. Ladies and gentlemen, please make sure the first step we actually do is always to identify, hey, this valve is between which two regions? I already chose the bicuspid. Bicuspid is between the... Very good. So just eyes on screen as well, LA, LV. So if we talk about bicuspid valve is between LA and LV, now I look at my graph, I must identify which line is LA, which line is LV. The two dashes. The long dash and the short dash. Can you see that? Okay, so now, if we choose to talk about close, based on this wrong direction, then we look at my LA, LV now. Let's say I want to draw an arrow. Valerie, is this wrong direction or is this wrong direction?
The second one.
Tell me the letters.
What? Wait.
Close means blood trying to flow in the wrong direction, right? Is it LA to LV or is it LV to LA? Thank you. So literally what I'm going to do here is draw this. But now, I will therefore complete it with... What does the big letter P represent? Look at the y-axis. Pressure. So now, we're done with our explanation already. Look at that. We've done our analysis of the graph. The bicuspid valve. Come, let me eyes on the screen. The bicuspid valve closes when the pressure in the left atrium becomes smaller than the pressure in the left ventricle. So we look at the graph then. When does pressure of LA become lower than pressure in LV? So like I said, this graph, look at intersections, isn't it? So therefore, if I do this for us, ABCD, at which point over here does LA pressure go below LV pressure? So graph skills, look at your graph. Can we identify when does LA pressure go below LV? LA below LV pressure. At which point here? EOY? Come on, name then think. Hey, don't wait for answer. Valerie, what do you think? B? B is a solid line is what graph? Yeah, nothing to do with aorta, isn't it? Look at that. Exactly, Rachel. Thank you. Point A, look at point A. This is LV pressure. This is LA pressure. Look at that. At point A, your LV becoming higher than LA one. Can you see that? So therefore, it is here where the bicuspid closes. Can? So we understand this is for point A. What is the question asking of us? Climax. And instead of analyzing every intersection, can I show you a trick to halve it? If this intersection close, when do the two graphs intersect again? Think first. Davina, when do the two graphs intersect again? When? A again? When does this line and this line intersect again? Don't look at me, look at the graph. It's there. I need you to think very properly. Look at the graph. Look at me, look at mine now. This is A. This is the LV graph. Oh, intersect one time. Intersect another time. Yeah lah, A and D. So if A close, D leh? Oh my god, this is the simplest question ever. If A close D, open. Thank you. Because the pressure is now opposite ma. At D, what happens? At D, what happens? You have pressure in the LV now becoming lower than pressure in the LA. You ask yourself, hey, this arrow direction, is this correct blood flow direction? LA to LV, is it correct? Yes or no? If you need to look at the diagram of your heart, LA to LV, correct or not? Yes. If not, you can think about the alphabet, right? Letters A to V. So A to V is correct. So if this is correct direction, the valve open or close? Open. So therefore, close, open. So now you look at this carefully. Intersection D is here. Time X is here. If here, then open. X is what? Here, then open Then before that at X close now. Can we understand this graph? So at time X, your bicuspid is closed. And apply the same logic to the aortic valve. Apply the same logic to the aortic valve. Aortic valve over here. Aortic valve. Between where and where? Aortic valve between where and where? Thank you, Leroy. Between the LV and the aorta. LV and the aorta. So, LV and aorta. I mean, if you already say this one is for bicuspid, then it has to be these two, ma. Correct or not? Then what's the closest one? If this is the intersection of C, then we find here lah. This point is what? Who become higher than who? You all learn how to read graphs properly. Can you tell me at point C here, who is becoming greater than who? Sorry? The left ventricle is this one. The aorta is this one. Whose pressure becoming higher than whose?
Oh wait, no, I don't know.
The aorta is going higher than the LV. Do we see that? So this one something we must be able to see properly. It's a very common error that we are making here. Can you see? All right, thank you for working on this with me. So therefore, at this intersection of C, what is happening is that aortic pressure becoming greater than left ventricle. So if I put my big letter P now, like this, hey, look at this arrow. Correct or wrong direction? Wrong. So valve open or close? Close is open or wrong is open or close? Do I want blood to flow in the wrong direction? No, right? So the door has to close now. So then you have your answer already. Has to close. That's it. Okay. Awesome. So I really need us to learn how to use this skill that I have just shared. Identify the chambers. Identify your directions. Ask yourself, correct or wrong. That's all. Okay. Hopefully this helps. Let's move on. Doesn't even have to be athletes. You and I, after chasing a bus, after finishing our 2.4, we stopped running already. Why are we still panting like mad? Why are we doing that? What's that for? To clear that oxygen debt, right? So what is the oxygen debt for? Breakdown. And that is where we have our answer. Okay? That's it. Well done, Livia. What is the working word we use to remember the function of nicotine just now? What was the first thing we highlighted about nicotine? addiction, right? It's called addiction. It's an addictive agent, right? Nothing over here about addiction, right? So A and B out already lah. Right now, look at that. So between C and D, tar. What does tar do? Remember this thing? Then tar. That's it. You have your answer. Okay. So emphysema is the bursting of the alveoli we talked about just now. Okay. It should be in your textbook somewhere on. So look at it. All right. And we talked about carbon monoxide over here. How does carbon monoxide result in these symptoms? So why will carbon monoxide poisoning cause headache, dizziness, confusion, or even death? What does carbon monoxide do? But we don't have that exact word there, right? So which of our four options here is the best one? Brilliant. And it binds more efficiently to hemoglobin than oxygen does. Okay. All right, we're going to do this very quickly here. This is the blood clotting one. All right, I've chosen to put this question down over here because it is a common one in which we are asked to identify structures in a diagram, similar to our first question in an MCQ. Okay, and then let's just verbally talk about this one and we can move on very quickly. What is cell G? Can you identify cell G? Based on its shape as well as what it's doing, what blood cell will not give you any marks at all? It's the phagocyte, right? So if we learn the specifics, we need to list the specifics. Okay, Olivia? All right? Because of the lobed nucleus, isn't it? You can see the lobed nucleus there. So G is the phagocyte. What is the name of blood vessel E? The capillary. Why is E the capillary? How do we know? All right, we have the wall that is 1 cell thick. The wall itself is 1 cell thick. We can also see that the lumen is 1 cell wide, isn't it? Only one red blood cell can flow through this at one time. And actually, that's what part B is asking. That's what part B is asking. 1 cell thick wall, 1 cell wide lumen. Easy. Okay? Quickly scribble that down at on the side. Let's go move on. Scribble it down because the answers will be given to you at the end also. So it's more important for us to know how to tackle the questions first. Okay? And you sure it's on the right idea already? Because what can we do about part C? You sure just now you're referring to something, right? For part C. Were you referring to something for part C? Oh, I thought you were looking for that. Okay, for part C, look at that. Describe the events that occur that effectively destroy bacteria. What is happening at F? Do we see what's F? Can we identify the cell there first? Very good. Lymphocyte. So just now, I shared with us the lymphocyte is thing of the archers on the castle wall, right? What are the arrows that they're shooting called? Antibodies, right? So that's it, antibodies. It's in our notes, right? So what do they do? Release antibodies. Then H, what is happening? The engulf and digest. Thank you. So it's all in our notes there. Okay, right? So just refer to the front. This is a very simple recall question. Every single mark over here should be easy in the pocket. Okay, right? So let's move on. Let's identify E and F. E is pointing to emptiness. F is the circled parts there. What's E? Wonderful, Xingyu. E is the right atria. What about F? Look at the circle. Look at the circle. F is the circle thing. The tricuspid valve. Thank you, Olivia. Okay. This is the RV, if the label says. This emptiness is the RV, isn't it? Okay. All right. Well done. Okay. Question is just simply, what happens? That will result in blood flowing. This, then this. This is from RA. to RV, isn't it? So stage 1. RV. Stage 2. To where? RV to where? Pulmonary artery. Aorta is for LV. Can? Aorta is for LV. And that's it. Why will blood flow in this direction like that? Why will blood do this? What's the RA doing? What's the RA doing? Why will blood do this? This morning you all brush teeth. All of you brush teeth. The toothpaste, will it just suddenly come out of the tube? You did something, right? What you do? Yeah lah, you squeeze the tube, right? So therefore, the RA contract, forcing blood into the RV. Fair? This one, oh my goodness. Correct direction of blood flow. Then, when I ask you this, something else comes to mind. If direction of flow is correct, then the valve open. The valve, no marks, how? The tricuspid valve opens. Can? Let's jot down the points together here. We'll do. Okay? So you talk about what color is blue? So in stage 1, in stage 1, RA contract, force blood into RV. Tricuspid open. Can you all formulate what to write for S2 now? Look at your diagram. See, I have given you 3 parts. I give you a few seconds to try first. Rachel, can you tell me what are the 3 points we write down for S2? RV contract, very good. Force blood into pulmonary artery. Last one. Very good. Pulmonary valve opens. Okay. So RV contract. Alright. This will give us 2 marks. How many marks is this question worth? Then how? Where's our 3rd mark? Since we talked about everything already, right? Or have we? Missing something. Question got 3 marks. We wrote down 2 only though. Come, where's our 3rd mark? This is what we're working for, isn't it? Everybody can get the first 2. Where's our 3rd mark? You don't know, you hear me?
He knows.
Faster. Come, where's our 3rd mark? Maybe your mother come to tell you answer. How is our 3rd mark? The valve. Which valve? The tricuspid. What happens? 2. Wonderful. Well done. That's our 3rd mark, isn't it? Look at that. When the RV contract, then how? Look at this. the tricuspid closes to prevent the backflow of blood into the artery. Well done, Cindy. No, it shouldn't be in orange. It should be still in green because that's for the S2. The door, please. Yeah, y'all know lah. Well done, Cindy. Thank you. Everything okay? Everything okay? I'm gonna miss you so much, eh? Can make sense, Learn how to find your points. Very good. School in Queensway. Your school is Queensway. Where is it? Queenstown.
Commonwealth.
Oh, so far away. You'll stay there also. Near there. Then when you come here.
So only time for like this one. Friday or not good.
Oh, thanks for making the journey here. Okay. This one. There's just one word I need you all to highlight. Makes the whole question easy peasy. Lemon squeezing. What word would that be? Thank you. Exchange. What do we get to exchange? Movement of substances. Surface area to volume ratio. Diffusion distance. Thin film of moisture. This all listed in the beautiful table I gave you earlier on. Can? So this one, let's move on. Very straightforward. Let's go. Lactic acid is something that we'll talk about a little bit more. Lactic acid is toxic at high concentrations. Only produced when? During anaerobic respiration. When does anaerobic respiration kick in? When does anaerobic respiration kick in? There's not enough what? Why not enough aerobic respiration? Why can't aerobic respiration increase further? What does it need that's not enough of? Oxygen, right? Oxygen. So over here, Q and P, who do you think is the more conditioned, the one that has a greater aerobic capacity athlete? P, right? Because P delayed lactic production. Okay, so for this amount of time here, P continued to undergo aerobic, whereas Q already started anaerobic already. So part one is just graph skills. Read the graph. They are telling you already. Increase sharply. Then just went off. Then you just take your pencil ruler. P is this one. Q is this one. Can? But if I just write down 10 and 12, 0 marks. Why? Thank you. Units are important. Right? Kilometers per hour. Right? Now, for part 2, describe and explain. For description, please take note. Trend data. Even if the question doesn't tell you based on the figure, based on the data, based on the graph, if the question is accompanied by a graph, use the graph. Do not throw away marks like this. Okay, describe the change, meaning, take note, only P only, don't need to look at Q, which means describe, 1 mark. As in first part, second part, the increase, isn't it? Relatively constant, the increases sharply. Okay, relatively constant, that's trend. Trend must always be accompanied by data. So what is this data here? What is this? Gradually or small gentle increase from, what's this? 1.8. Alright, read your graph properly, 1.8 to 2.1. Alright, from 4 to 12. And then sharply increases 2.1 to whatever that value is, 10. From 12 to 20. Is it 20? No. 19. 10 to trend and data. This one, I shouldn't have to teach us to read graphs already at this point. What I do need to do is remind you to be careful in reading your graphs. Alright, so usually errors from trend questions come from carelessness, not looking at the squares properly. Clear? Right? And then the explain. So this part, why barely increase? What is this person still undergoing mostly? Mostly aerobic. That's it. This part, why like that? Mostly aerobic. Second part, why increase drastically? Anaerobic lor. Can? Anaerobic. Easy stuff. Okay? Come, what are we looking at here? What are we looking at here? I have cells J and K. Look at what cell J has on top. Cilia. Alright. But Olivia, that is an extremely common overlap that we make. So please make sure they look kind of similar. That's why you were thinking of villi there, isn't it? But villi is found where? Small intestine. And function of the villi is for absorption. Okay, so listen up. Livia will definitely not be the only one that makes this mistake. So therefore, after today, let's try to be not making this mistake. Villi and cilia, they look very similar because they're just like that. One is to do this, one is to just stay there and absorb things. Clear? Alright, thanks Livia for letting us discuss this point here. So, but J, take note, J is not cilia. What's the name of cell J that has cilia on top of it? What's the name of cell J that has cilia on top of it? It is not cilia cells, unfortunately. It's not as simple as that. Flip through, where do we find it? What is name of cell J with cilia? Flip earlier, backward some more. Ciliated epithelial cell. Okay, so I'm gonna write it down together. Practice your writing. Cell J is ciliated, like got cilia lah. Can epithelial cell tells us where is this cell on the epithelium. It's the wall, referring to the wall. Can? So what's K then? K is much simpler. What am I drawing? Mucus. So cell K, mucus producing cell. Okay, I give you one difficult one, easy cell ma. Okay, so therefore cell K is the easy one. Cell K, mucus producing cell. So we talk about their function of protecting the human respiratory system. Whose cell's function should we talk about first? K first. Why? Remember the idea I asked us to sweep there? If you talk about J first, then what are you sweeping? Then if you say, oh, to sweep mucus, then where did mucus come from? You could do that. Or if you talk about K first, you immediately produce the mucus already, trap the dust already, then cell J comes in to sweep the mucus away, isn't it? Right? So if you are able to sequence our thoughts like that, it makes our life much easier. It makes our answers much shorter as well. Okay? Right? Because Livia, if you talk about J first, absolutely fine. Just that later on, you might have to add in a bit more words to connect the 2 points. Right? You can still get your answer, yes. So what am I suggesting here? If you can think through it properly, in terms of our explanation, which one should come first? Hey, the mucus one should come first. I produce the mucus already, trap the dust and pathogen already, then the cilia comes in, do its magic. Can? And always, we are sweeping away from the lungs towards where? The throat to be? Or expel, thank you. Okay? And with that, we're done by a portion. Any questions? Okay, so if not, let's go on to the chemistry portion. Let's go on to the chemistry portion. And right off the bat, we're gonna put a nice little cross on salts. Okay, salt preparation specifically by itself, not required in our syllabus. And in fact, within the mole concept, acid bases, and QA, there are some things that we are removing as well. Yes. Oh yeah, come. So, okay. Only she doesn't have. Oh. Okay, and even within 1, 2, and 4, there are things that we will remove as well. So if you see this, and then I move to the next page, yours don't have, you will see the thing written there as well. Not required. Let's go. So, similar idea as what I have shared earlier on with the bio parts there. If you are tested, that's great. I'm going to walk you through it a little bit. If you're not for your Sept EOY, we are working towards our O's next year as well. So work on this with me. It is unlikely that you'll be asked to define AR. But it's still important for us to understand what it means, isn't it? Okay, because when you put down AR and MR together, right? MR is the sum of ARs. If I just keep it like the simplest, MR is sum of AR. And what is AR? Sum of nucleons. Wow, nucleon. What topic we learn nucleon at? What are nucleons? What are nucleons? Nucleons are just like Singaporeans that live in Singapore. So nucleons are things found in the nucleus, but not of a cell, of an atom. What's found in the nucleus of an atom? You learn 3 subatomic particles. Electrons, protons, neutrons. Nucleons are protons and neutrons. Wow, you all like that? A bit worries me a little bit, but okay, let's go in together. All right, so basically the mass, if I want to look at the word mass, or AR, okay, what does it tell me? F then nucleons, how? If we expand upon nucleons, do these symbols make sense? Yes or no? Yes or no? No, What is the thing I just removed from P? Positive, right? What is the subatomic particle that has a positive charge? Hey, P for positive, P for proton lah. Correct or not? Then what was the Livia, what was the symbol I removed from the N? 0, because neutral charge, neutron. Can? Alright, so protons, neutrons. Yes, of course, go ahead. Are you feeling okay? Okay. Alright, so these are the nucleons there. What happened to our third one just now? Rachel mentioned, right? What was it? Be familiar with these 3 symbols, okay? Right? As we move on in our syllabus, we will use them more as well. The proton neutron found in the nucleus, so they called nucleons. Where the electron? Around the nucleus, right? What's it called? Shells. Thank you. Right? The electrons are found in shells. Oh, come on. Okay. Let you in on a little fun fact, right? Chem was actually the first science that I fell in love with. But I'm mostly teaching bio now also. Like, hey, why you give me that face, Valerie, Right? Hand one, work on this with me, alright? So, if I know that AI is the sum of the neutrons per atom, then MI is what? All the atom add together, lor. Okay, just sum only. Okay? Right? So therefore, here is MR is it's the sum of AR in the molecule of the compound, lor. Right, some quick counting. Suhan, water molecule formula. The letters and the numbers are there in the wrong configuration. HO2 means 1 hydrogen, 2 oxygen. If that was water, H2O, 2 hydrogens, 1 oxygen. Okay? So, hydrogens AR, listen, you all should be able to do this very simple mental sum. Hydrogen AR is 1, oxygen AR is 16, water MR is H2O, 18, thank you. H is 1, H2 is 2 lah. Then oxygen 1, 16, 2 plus 16, 18. Okay? Carbon dioxide formula? CO2. Listen, carbon 12. Oxygen 16. CO2 is? Very good, 44. 10? Alright. Shall I challenge your one last one? Then can write down if you want. Write down formula of calcium carbonate. Calcium carbonate formula. So even though you're in chemistry, right? Even though at the end of year, the list never put chemical formula equation, right? Confirm will be in. Because it's a cumulative thing. CaCO3. Okay, write that down. CaCO3 is calcium carbonate. Okay, CaCO3, calcium carbonate. Seeing your periodic table, this reminds me, right? So for those who join my classes from next year onwards, right? You'll be given a nicely laminated one. Ivy, can you show off a little bit for our friends here? Hey, higher lah, I must flex a bit. Okay. So that laminated one, right? Front got periodic table and back is littered with lots of other information as well, including our mole stuff. Okay. Right. CaCO3, calcium is 40. Carbon is 12. Oxygen is 16. What's the MR? That's very easy. It is 100. Very good. Okay. Wow, so fast. Oh. How you get 74? You miss 1 oxygen. No, 2. Is it?
Times 3.
CaCO3 40 plus 12 plus 16, 16, 16. Oh, you use 20, is it?
I use 14.
Okay, so it's 100, right? Okay, hey, please, do not need to memorize all these things, okay? It's just a little fun thing for us to work on together to be able to count and use our calculators properly. Okay? Come, let's get back to this. So you realize that relative molecular and relative formula mass, they actually are the same things. But term-wise, wow, shudder this Alright, but the names are different, but the way you calculate is the same. Molecular mass for molecules, formula mass for ionic compound. But the way you calculate it is the same. Okay, so just now, H2O and CO2 are molecular masses. CaCO3 actually is formula mass, but the way we calculate it is the same. Okay? Let's continue. Molar mass is when we bring in mole. The idea of mole, if you are having trouble understanding this, let me refresh the perspective for you. What number comes to mind when I say a pair? 2, thank you. What number comes to mind when I say a dozen? What number comes to mind when I say a mole?
24.
Where did 24 come from? It's a long thing with the standard form one. Avogadro's constant. 6.02 times 10 to the power of 23. Do you know what times 10 to the power of 23 refers to? You put like 23 zeros behind decimal point. Huge number, isn't it? Shouldn't you understand that? Yeah. So it's a huge number, right? So to make it simpler, we chunk that thing as 1 mole. Can? So if you're having troubles understanding that, I hope it's easier now. It's the same as pair, dozen, triplet, all these kind of things. Okay? So, couple of things for us to cross off. The entire bottom row, not required in our combined syllabus. Wow, satisfying. Wow, okay. Alright. So for us, you just focus on those at the top there. Please keep in mind, the word particle here is extremely generic. Particle refers to anything. And look at this, etc. Right, 1 mole of humans lah. You know what's the global population? What's our global population? In the billions only, right? Billion is bigger or smaller than mole? Way smaller. Way smaller. So I want you to even like appreciate like, oh my goodness, how much a mole is, isn't it? Okay? Alright, so coming back over here, Avogadro's constant, please do not confuse this with the fruit. Okay? You will give your teacher a good laugh, you will avocados constant, okay? So Avogadro's, right? It's named after a person, not the fruit. Okay? So we use that to find the most important thing for this topic, number of moles. Please remember, you only have 4 formula to remember. Do you all learn the triangle method of looking at this? You all learn in math also, the speed, distance, time thingy. You use that still in math? Then you all use that triangle for this. Yes? No? You don't even know what I'm talking about? Not the liver anymore. No more triangle. So let's write it down together. Okay, we have a little bit of space at the bottom, so let's work on the first one. This one over here. Okay, you know what? I'm just gonna number them like that. 1, 2, 3, 4. Okay, now I'll draw my triangles respectively. Can? Triangle 1. And I'm gonna cheat a bit. Oh no, no space. Oh no, look, I'm drawing some cult thingy. No. How do I separate it? Okay. Okay lah, good enough. Okay. Okay, first one. I will start off with this. Then you look at the printed box there. What do I put at the top and the right? On top of what? On top of what? Over? Molar mass, right? This one, I will write down AR or MR. This one, I need to put bracket. Wow, leave your triangle so small leh. Triangle draw bigger. Okay, so the couple of things here to take note of. This one must be in grams. Sometimes question give you kilogram or must convert. Can? But in grams means like mass, right? Wow, what if they give you volume? Can we convert volume and grams? That's volume, ml and grams how? Some of you may have it in your head that 1 gram kind of same as 1 ml, right? But in chemistry, when you're doing like precise calculations, cannot. So then if you're given volume, how? We calculate it another way. That is for number 2. So it is still mole. You give me volume, how? You give me volume, how? I will divide it by what? Look at box 2 lah. Very good. What does dm stand for? Alright, D, decimeter, but I mean the name doesn't matter. What's more important is how to convert to something that you're more familiar with. Instead of dm cube, you're more familiar with cm cube, right? So how to convert? Then 1 dm cube is to 1000 cm cube. So this conversion is super important. As long, then this volume one, what units must it be? This one must be in grams because this one will be based on grams. But this one, if the question gives you cm cube, how? You either convert this to cm cube or convert this to dm cube. As long as both sides the unit same, it works. You understand? Yuxuan, if the question gives 406 cm cube, can you divide directly like this? Do what? Change which one? No, Yuxuan. Change which one? 406. What do I do to it? Look at this. Yeah lah. So you either change the cm to dm, or this one you can change to cm also ma. How to change that to cm? Cube. Yeah. Can? Either way works, as long as both are in the same units. Clear? Let's go. You will realize that I always start at the bottom left. Concentration in mole per dm cube. Concentration. Can we think of concentration as crowdedness? Number of people per space. Can? So for example, now we have 2, 4, 6, 8, 10 students per classroom. So our concentration will be 10. Can? So this one is actually the easiest triangle. It is based on the units itself. How do we read this slash? Per, right? And in math, what is per? Divide. Per is divide. So what do you do? Mole per dmq. And dmq is a unit for what? Quantity. dmq is a unit for what? Quantity. I'm looking for, is it length? Is it height? Volume. Can? Last but not least, this one. Let's see whether y'all can work it out. Can you use triangle 3 to help you? Leroy. EOY. Then what to put here? Irie. Mole? Mole per dm cube. Mole divided by dm cube. Gram per dm cube. It's the same. 3 and 4. Look at that. So then these 4 triangles will last you through your entire mole calculations. How do I use the triangles? If you want to find volume, you cover volume. Then what do? If it's side by side, it's times. If you want to find volume in dm cube, you cover volume in dm cube. Top and bottom means what? Divide, no? That's it. Can? But before I move on, triangle 2 got one important limitation, which is a good thing also, actually. This one is only for gases. So the word volume here, only for gases, cannot be liquid or aqueous. Can? So actually, your syllabus for mole is 4 triangles and one concept here, limiting access. Let's move on to the next page. I'll find limiting access wherever that is. And you have to highlight this thing for me, please. With emphasis on the word completely. With emphasis on the word completely. Why is the limiting reactant, why is the limiting reagent so important? Because and if you're wondering what yield is, amount of product that forms. So this is why I say, even if your EOY doesn't state chemical formula and equation, still need what? You cannot go anywhere without formula and equation. Can? So this is the reaction between name Xingyu. What is this? Yeah, aluminum carbonate. Can? Valerie?
Sulfuric.
Decide. Huh? Which one? Sulfuric acid. Very good. Okay. Livia? No R. Aluminum sulfate. Can? Aluminum sulfate. Water carbon dioxide gas. Okay. This does the reaction there. Question is saying what volume of this concentration of the acid you require to react completely with 234 grams of aluminum carbonate. The way we look at this here is always learn how to determine which number to use first. And the only reason we have to determine this is because it gives me information on more than one reactant. And that is actually your first clue. Hey, Joe, when do I need to calculate limiting access? When they give you numbers of more than one reactant. If they only give you a number for one reactant, just go straight to the calculations. But because they give me both, I must tell me which one should I use. Okay? Yes. Ask me, why ask her? Ask me. Come, quickly. You don't ask me later, I'll ask you more. Okay. So 234 grams of aluminum carbonate. What can I do first? What's triangle number 1? Because what's triangle number 1? You better be more present. We drew triangles just now, right? Just now. Yeah. What's triangle number 1? What is that for? Can you just tell me what triangle number 1 is for? What does it allow me to calculate? Number of moles. And how do I do that? I take the mass, divide by. I need to think before you answer. To find the number of moles, I take the mass and divide it by. Or read the whole thing, okay? Don't need to word by word. I must prompt you. Okay, Nathan? And that's what we're looking for. I find the MR first, 234. And just 234. Sometimes question will be very nice like that one. Can? Okay. So 234 grams over 234 in MR, I get 1 mole. So I have 1 mole of sulfuric acid here. However, what does this big number tell me? 3 times in terms of the ratio. For every 3 sulfuric acid, it requires. Sorry, what did we calculate? We calculate for aluminum carbonate. So for every 1 aluminum carbonate, I need 3 sulfuric acids. So if this is also just nice 1 mole, then the ratio over here is I need 3 moles. Can? So by mole ratio now I need 3 moles of this. So now I have number of mole as well as concentration. Question asking me for? Read the question. What's the question asking me for? Volume. Thank you. So based on these 2 numbers, can I find volume? Find your triangle. Which one do I do? Which one should I use? How do I find volume based on these 2 things? Which triangle has this concentration? Number 3, isn't it? Triangle 3, isn't it? So therefore, triangle 3 is mole per dm cube and mole. I have number of mole here, isn't it? I have concentration, isn't it? I have this and this. Can I find this? Divide lor. Divide. That's it. So 3 divided by 1. Done. Okay. So that is for the reactant side. Now question asks product carbon dioxide maximum volume. If one ratio equals to 1 mole, so now how? 1:3 again. So if this is 1 mole, this is 3 mole as well. Yes, Ari? And ask you something? Yes, Divya?
How do you get the ratio for the mole ratio?
In the topic of chemical equations, what do you do as well? That is the meaning of balance. If you all don't know, if you all haven't realized why we balance, is for this topic here. To give us a ratio. So we're not balancing for fun. I mean, sometimes it's fun balancing, right? Okay. Okay. All right. So the purpose of balancing is to get to the mole ratio, which helps us with the mole calculations. All right. Then again, see this one just now we 1:3 in the part A. Now we 1:3 again. So now I have 3 moles of the gas. But because it's a gas, something special we can do. Triangle 2. Share, you tell me what to do. 3 moles of carbon dioxide gas. How do I find the number of the volume of carbon dioxide gas? Volume. I want to find must times ma. Why not? So you just see them 24. That's all. Ta-da. Can. If the question asks you, hey, live in cm cube, Valerie, how? times 1000. Okay, easy. Let's go. But now, in a more realistic question, they give you information on more than one. What I like to do is color it accordingly. This one cannot see, huh? Okay, so now they give me information on more than one reactant, isn't it? I must find the number of moles of each. Look at the ratio. Hey, which one can be completely used up? The one that is with the limiting. And I use limiting for all my subsequent calculations. Okay? From here, again, mass, solid, mass over molar mass. Done. Okay? So this. See now the number? Not as nice anymore, isn't it? What does this mean? 5 significant figures for workings live in 5SF. Final answer? 3SF. We all remember this, but do we know why? Thank you for accuracy. Thank you. Okay, if you 3SF all the way, right, your final answer will be very far from the actual. Okay? Right, if you all got time, you all go and try one, then you do both lah. You see the numbers confirm different one. Alright, so 5SF for interims, 3SF for finals. Alright, so I find the number of moles of each. From here, I do this. From here, I do this. Okay? Now I see. My equation tell me 1:3. If I finish this one up, I need thrice of this. Sorry, I need this one times 3. If I finish this one up as 1 mole, then this one I need 3 ma. So if this one times 3, will I have it enough? For example, this one times 3, I'll get this. But I have this amount. Do I have enough? Are you able to see? I have 0.05. I need 0.03. Logical? Yes, so I can completely use up my aluminum carbonate. But that's my limiting already. But let's check for the opposite as well, isn't it? Because Yushen is like, yay, what are you talking? Okay, right? For example, Yushen, if this is completely used up, 1:3, right? This is 1 or 3? 3, yeah? So what is 1? Count for me lah. Calculator! Read out, come on, let's go. So I need this value. Do I have? Have or not? I need 0.0166. Do I have? Open your eyes, Yushen. I only have this amount. 0.012 leh. I need 0.016. So can I completely use up 0.05? For me to completely use up 0.05 sulfuric acid, I need 0.016 aluminum carbonate. Do I have? So therefore, can I completely use up 0.05 sulfuric acid? No. Definition of limiting is to completely use up. I cannot completely use up my sulfuric, so it is the excess. And so find the one that can be completely used up. Okay? So then from here, this will be the number of moles I use in all subsequent workings. So as you can see, I will always use times 3. So from here, at times 3, I'll get this. Okay? Alright, so always make sure if they give you more than one, calculate the number of moles of both, look at the ratio, find the one that can be reacted completely. Okay? Let's move on. So for gases though, can we see? Gases. There's something very special as well. there's something very special for gases as well. Volume ratio, we can equate it to mole ratio. Without any calculations, straight up equate. No need calculate. But now they give me grams. How to change? We cannot change. There's no conversion. You have to calculate. If they give me mass, what do I do? How do I find mole? Is there a triangle that has mole and mass in it? Absolutely, the first one, isn't it? So it's 28 times or divide? Times or divide? Come. 28 times or divide something to find number of moles. Divide, okay? And for this topic, please make sure presentation, very important. 28 is the mass. What's the MR? Find the AR of nitrogen. AR of nitrogen is 14. MR of N2 then is 28. Can? So therefore, it will do you good to just show it out like that. Okay? This one don't need calculator, right? Ah. This is where the thing that Livia asked just now comes into play. What's the mole ratio, Livia, of N2 is to H2?
1:3.
Well done. So therefore, this tells me equals. Tell me how to do it first. Don't tell me the answer. What's the workings? Well done. Then from here, they ask me this one. So I need to find number mole again. But before that, Sohan, tell me the ratio, please. Look at the equation above. How many nitrogen? How many ammonia? Yeah, two. Question you're asking for? Mass. I got mole. How do I find mass? Is that what you're asking? Oh, that one was also mass, Okay, yes, you're right. Come, let's do this first. Yes, you're right. Livia, mass of NH3, 2 times MR, AR is only for one thing. NH3 got multiple, right? So what is the MR of NH3? What's the workings here? AR of N is 1, 4. NH3. I think this one easy lah. Can put 3 lah. 10 turn into 1 times 3. Okay? Whatever this value will give you is our answer. Value what do we get? 34, very good. Apologies, yes, good catch, Rina. Question is also asking for mass, Then we do the same law, 3 times. What's the MR of H2? EOY. 2, very good. Can? So the second part over here, C and D, is where we use this magic. If it's gases to gases, volume ratio is the same as mole ratio, which means if you are given the volume of a gas, you need to find another gas. No need to convert. Just go straight. Question tells me 90 cm3 of H2 here. 90 cm H2 Question asking me to find volume of N2. This one. So the ratio here is 3:1. Alright. The ratio here is 3:1. Purple is 90. What's the value here? Exactly. Easy. If it's gases to gases, easy. Okay? Straight away like that. What are the workings then? How do we present it? Like I said, presentation is important here. Again, by mole ratio. I typically put the one that I want to find later in the ratio. Okay? But there's no right or wrong here. So this tells me 3:1. And therefore, 90 is 2. That's all. Okay? This part, if you want to show your workings, could be better as well. You will simply put then how much ammonia then? So what's the ratio again? 1:2, isn't it? Alright. So now if my N2 is 30, what can we do? We can do very good. And that's pretty much it for mole calculations for our syllabus. Who's tested on mole for your EOYs? Okay, only the 4 of you not tested. Can follow? Kinda. Excellent, thank you. Let's move on to acid and bases. Okay. Definition of an acid. Substance that releases what when dissolved in water? H+, fill that in please. We're gonna skip basicity. Strength is not strictly in, as you can see over here. But it is something important for us to look at because it comes back again, especially when you look at organic chem next year. Okay, so it's simple enough. Strong, complete. Weak, partial. These four words easy to keep together, right? So a strong acid completely dissociates. A weak acid partially dissociates. What does it mean to dissociate? To separate into ion. And to separate into ions. Alright, and so the words here, you can peg it together. Strong is complete. Weak is partial. How do we represent them differently then? The equation, it shows. Blue for strong. The standard arrow lah, the equation. If it's weak leh, reversible arrow. Keep in mind, the reversible arrow, if you write it down this way, why? What's the difference? The arrowhead must be half. Okay? Unlikely that you'll be required to do this, but it is a thing. Right? Cannot draw two full arrows. Must be half. Let's go. So because we already crossed out basicity, these words don't need. The mono and di don't need, but strong and weak still in. And okay. Base. How do we identify bases? This part over here, when we are learning this, I always do this. Momo. What's Momo? Irene, what's Momo? Yes. Then O leh? Take away the H lah. Oxide, yes. And so tell yourself that lah. Bases are Momo. What's the difference between a base and an alkali? Everything in this circle is a base. Everything in the green circle is an alkali. Which one is the bigger term? Base lah, obviously based on this Venn diagram, isn't it? So what's all alkalis are bases, yes? All alkalis are bases. Are all bases alkali? No, so there is a difference there, isn't it? So what makes this a base but not an alkali? A non-alkali base. So what makes a base an alkali then? Alkalis are momos. Alkalis are momos. What is that? What am I looking for? You know? Come on, come on. This is your clue. Soluble. Alkalis are simply soluble momos. Okay? Soluble metal hydroxide, metal oxide. Alright? Alright, so soluble bases are known as alkalis. So this is the opposite here. Acid is H plus. Alkali is OH minus. Thank you, Rachel. Alright. This part over here is the same. So no need to learn anything new here. It's the same. And definition for strong and weak is the same for acid and alkali. So equation, again, the reversible arrow appears as well for the weak one, and then the single-headed standard one is for the strong. Typical questions here about the salt too acidic, what can you do? Add the calcium hydroxide, add a base, simple as that. Okay. Alright, so these are scenarios and questions that we have worked on together in our worksheets, right, Tyree? Very good. I'll say remember. Tyree. Okay, so reactions. Very simple. For reactions here, please remember acid got three reactions. So remember it as MBC. Metal, base, carbonate. Acid reactions only got three. MBC. Metal, base, carbonate. Can? Right? So you have acid reacting with a metal, acid reacting with the bases. These are all bases, and then acid reacting with a carbonate. Right? So they're listed differently here. Doesn't matter. So all reactions will always produce a salt. Please remember that. All reactions will always produce a salt. As long as an acid reacts with something, a salt will be produced. Okay, a salt will be produced. So therefore, the acid will contribute its H plus to form the hydrogen gas. So it's from the acid. The acid will contribute its H plus to form hydrogen gas. But if now acid got H plus, yes. But the carbonate, what's the formula of a carbonate? It's CO3 2 minus. I want to urge us to just remember the sound. Carbonate, carbonate, then will produce carbon dioxide gas. No. But from CO3 to CO2, what's missing? The oxygen, right? So therefore, previously, it's just H2. Now is H2, but the O comes in also. So therefore H2O. Yes, go. Can? Alright, so it's very straightforward. Just think about what is present in the reactants. Okay, that's what's present in the reactants. Iron and lithium. What are some unreactive metals? It's three in your syllabus. Silver. No, cold water is neutral oxides, Irene. I asked you because reactivity series. Silver. Copper. Another jewelry metal. That's not a metal, isn't it? So it's just the metals below hydrogen in the reactivity series. Copper, silver. Another jewelry metal. Gold. Okay? Unreactive metals, copper, silver, gold. Alright, for the rest of you, they have learned it in reactivity series, that's why. Okay? Can I read it here? This one must remember, unreactive, copper, silver, gold. So literally, like, all metals will react with acids to do this, except copper, silver, gold. Or if the question gives you information, this one is below copper, silver, gold, below hydrogen, then it also will not react well. Because the reactivity series is a guideline only. It's not exhaustive. It doesn't include all the metals, isn't it? So it's a guideline only. Okay? Alright. Fairly straightforward stuff over here. What I can think of to remind you is, hey, spelling of effervescence. And whenever a gas is produced, please make sure to include the appropriate observations, colorless and odorless. Right? Obviously, if it's colored and got odor, you say lah. Alright, last one. What's the name of this reaction here? Neutralization. Thank you. So the acid is neutralized, the alkali is neutralized. Look at this. Temperature of mixture increases. Neutralization reactions got no observable change. Why? You cannot look at the reaction and say, okay, it is occurring now. Why? You look at the previous two, look at this. In the acid metal and the acid carbonate, gases are produced. And you see bubbles. But for neutralization, you cannot see. So how do we tell? Stick a thermometer in and we tell the change in temperature. Can? So over here, they're asking us, hey, there's a note here. If an acid reacts with an aqueous ammonia, so we will have the Alright, so there's something unique over here. Alright, and last one over here. Your reaction doesn't involve an acid, it's an alkali already. So alkali plus ammonium salt, you will have pretty cool one. Pungent gas. Anyone smelled ammonia gas in the lab before? Anyone had the opportunity to smell ammonia gas in the lab before? Well, we look forward to the day that we get this pungent gas then. Okay, right? It is not like super strong, but it's definitely not something that you can live with for more than a few seconds. Okay, all right, come. So these are just the 4 reactions. It kind of looks like 2 over here, but you can clump it together. So 1, 2, 3, 4. Okay. For oxides, okay, now Irie, you get to tell me that. What was the thing that you said just now? No cold water, the neutral oxides. Now, why is Irie telling us no cold water? Because examples of neutral oxides that do not react with acids and alkalis, you literally write it down as you have heard it. No cold water. What does no represent? What does no represent? It is an oxide. What is this? Nitrogen. So what is the name of this? Nitrogen oxide is incorrect. There you see? What's the difference between these two? So you must say monoxide to represent specifically one oxygen there only. Can CO2, carbon dioxide, CO, carbon monoxide. Likewise, nitrogen monoxide. Can? Right? So actually, do we see your neutral oxides or your monoxides? One oxygen only. Can? And because they are neutral, no reaction. So this got double meaning lah. Alright, no cold water to help us remember, as well as no reaction with acids and alkali. Okay? Alright. So therefore, all non-metal oxides are acidic except for no cold water. Remember this sentence. All non-metal oxides are acidic except for no cold water. So let's use the same thing over here, isn't it? All metal oxides are basic except for something electrifying. Remember something electrifying, Ari? Those are lead acid bases. How do we remember the amphoteric oxides? What are the 3 amphoteric oxides? What do you remember? Come on, come on. What are the 3 amphoteric oxides? I don't think you can find it anywhere. Just gotta remember it. How safe about it? Good try. You all learn it now, acid bases. Then the amphoteric oxides are what? Oh no.
Zinc oxide.
Yes, yes.
Lead.
Zep. Zep. The Zep oxides are amphoteric. Zinc oxide. Aluminum oxide. What's P? Lead. Thank you for not saying potassium. I just met up with some of my Sec 4s that are now in Poly 1 until today. She keep telling me potassium. So therefore, please help me to remember the P is for lead because the symbol for lead is Pb. Can? I mean, I could say ZAL, but doesn't have the same ring to it. Zal. Okay? All right, so these are the four oxides. Please, you'll do you good to remember them. Acid bases done. Now for QA, let's move on to QA. QA. Now QA is a topic that we develop a love-hate relationship with. Love because QA, qualitative analysis. Now love because very little to learn actually. Very little to understand. And that's also where the hate comes from, because it is it's just simply one of those topics where I have to just say, please remember the table of the color changes, what happens when you add the substances. For example, if you look at QA, what are some of the few things that I can like teach to help us understand is your test for cations. The test for cations, listen up, it's always a two-step process. So if the question doesn't give you any indication of a two-step, dropwise first, then access, it is not a cation test. So use that to help yourself. Oh, add a few drops first. Observe, add access. Observe. That's your cation test. Okay, what are the two reagents we use for our cation test? Sodium hydroxide, aqueous ammonia. Hey, both of them are actually bases. They're both alkalis. Sodium hydroxide is a strong alkali. Aqueous ammonia is a weak alkali. So, you can see how we have broken it up. White versus color. The color is the easiest. You recall, like, maybe even Sept 2, you all did some Chem experiments. You all created some crystals. Oh, do you all remember the color of the crystals? Oh, you made white one, so boring. What other colors do we have? Light blue. Pink, you say pink? Wow, pink would be cool then. The light blue crystals, they did in crystallization, separation techniques. That would be crystals of copper 2 sulfate. So light blue is for copper 2. Okay? Reddish brown is for the color of rust. I'm sure you've all seen rust before, right? So reddish brown is the color of rust. So iron 3 is rust. And then iron 2 is light green. Okay? This is just a very brief, concise summary of the QA table there, right? So either look up your own school notes, or either you can look at our notes there. It's the full table there, a bit clearer to see. All right, but if not for this QA part, right, nothing much to look at except for example, cation is step 1, step 2. Okay, and then I have two sodium hydroxide or aqueous ammonia, right? There are two very different ones. All right, so this is for you to go and look up on your own. Keep on referring. I do not recommend you. Wow, okay. After you look at it, okay, test me. No, don't do this kind of thing. Keep on using it, keep on referring. Familiarize until memorize. Okay, so for the yeah, same. Okay, anion test then. You can see we are not going to be required to look at the iodide. We're not going to be required to look at iodide, so you can cancel out the iodide. Okay, you can cancel out the iodide. But if we see barium, jump straight to testing for sulfate. When we see silver, jump straight to testing for chloride. Okay? Right? These are the two things that we're looking for. These are the two things that we are looking for. And then this one, nothing new. Look at that. We learned this since lower sec already, a white PPT. Okay, we look at our white PPT thing. Last but not least, this one is simply production of the alkali, the ammonia gas. All right, so we look at what jumps out at us. Wow, aluminum foil. So whenever you see the reaction got aluminum foil, right? That's for nitrates already. That's it. Okay, so if you have this one, oh, test for carbonate. When I use barium nitrate, test for sulfate. Use silver nitrate, test for chloride. So these are the things that I can point out to you to add these little droplets things to help us remember things more easily. Okay, so there's really no use in us blindly trying to cram the entire table into our heads. Look for these tiny little points. Last one is a test for gases. I think this one is the easiest because we have all done at least the carbon dioxide one before, PPT. We have all done the hydrogen and oxygen one before. Okay, what will be new? We have done this in the acids topic as well. Okay, because ammonia is an alkali, isn't it? So what will be new literally is that the two that we haven't talked about. We have chlorine gas and sulfur dioxide gas, both of which are very toxic. Right? Both of which are very toxic. Chlorine gas and sulfur dioxide gas. Chlorine gas is a very unique color change that we need to observe here. And it cannot be simply red to bleach only. The test for chlorine gas must begin with blue. You must state blue to red to bleach. It cannot be just red to bleach. Okay, so if you are suspecting that something is chlorine gas, you go and use red litmus paper. Oh, turns bleach. You write down red litmus paper turns bleach. No marks. The full observation for complete identification of chlorine gas is blue to red to bleach. Okay. As for sulfur dioxide gas, what's the name of this thing? We encountered this before. Alright, so this is something that's super important. We will encounter this in a later topic called redox. Right? For now, KMNO4 works. KMNO4 works. Right? The proper name is potassium manganate 7. But for now, its original color is purple. It will simply turn colorless if a pungent sulfur dioxide gas is present. So actually, of all these things, right, color and smell will always be your primary observation. Always go with color and smell first. Because if you think about it, these three, then you can test for them, right? But if it's colorless, yet pungent, ammonia gas, but never go with your suspicions, do a test. So now the color's gone already. If you got color, how? There's only one in your syllabus. Got color and pungent. Alright, so actually these two are the ones that we compete. Then last one is simply but colorless pungent. Why never put? Colorless pungent also lah. This one also same. Okay. Alright, that's it. Right. QA, very straightforward topic. Like I said, it's a love-hate kind of thing. Right, it's very straightforward one. Just gotta work through your tables. And okay, that's it. However, if you may realize, time goes by very quickly. We're having so much fun, isn't it? Okay. I hope that was a snigger in a yes acknowledgement. Okay. As I shared earlier on already, answer keys will be given, right? But for this one, maybe we can just look at one of the questions that we can work on, especially for the mole. I think mole is something important. Let's look at question 2 actually. Let's look at question 2. We did a couple of exercises in mole just now. So question 2 will help us with this. We're gonna use the same methods. For example, I'm gonna color code so that my numbers are easier to see. Okay. Which reactant do I have a complete picture of? Which reactant do I have a complete picture of? Green or purple? Pink or purple, whichever. Okay, so that's one. So I have volume and concentration. What can I do with volume and concentration? What can I do with volume and concentration? Number of moles, thank you. Volume and concentration, what do I do with them to get the number of moles? Can I just do this? Why not? Alright, so my 20 is in cm cube. I gotta convert it into dm cube because my volume in the concentration is dm cube, so it has to be. PPT tabs on your calculators, please. Valerie, thanks for calculating that for me. Sorry? Absolutely 0.
0.02.
Thank you. No fractions in mole. 0 point. Thank you. Okay, I have that. But this one is not a limited access thing. They already tell me exactly already. So how? Concentration of sodium hydroxide. So if I mole ratio this one, how? Livia, do you know what I'm looking for?
One.
Is to what first? And my ratio is? Well done. So this brings me to I have number of mole, but the question is asking for. Thank you. How do I find that then? Hey, wait, I just realized this one. Not potassium, not sodium, potassium. Okay, what's the concentration of KOH then? What do I do? How come, especially those that are doing this for EOY, come on. What do we do? Vision. Divide by volume, so 0.04. Like this. Then how? Tell me what to do instead. I don't need the number. Okay lah. Huh? No, not 1,000. Divide by 1,000. Okay, convert to DM cube, 1.6, thank you. 1.6?
0.00.
Who tell me 0.02 just now? Was it bad?
No.
I got scammed. So this one is now then we have the correct answer 0.160. Yes. Okay, see, that's why I tell you, it's important to have your scientific calculators with you. So thank you for those of you who brought it here, right? For calculator, sometimes we'll still press wrong, see wrong one. So we gotta learn. Okay. Okay. The rest of it, right? Please make sure that you go ahead and try it, right? And when you get the answer keys, go ahead and check it out there. I wanted to focus a little bit more on the going through the content there, so that I can make sure that I can help us to learn the stuff that we may have missed out in school. So I hope that you have found that useful. And before I lose all of you, can I ask you all to do me a favor, please? Right? Just do a quick scan of the Tampines one over there, because we are here at Tampines. And you can see the beautiful foolscap pad you have over here, right? If you tell me you got a lot of paper at home, right? Sure, right? But it is still something that you can get over here. Who doesn't want more paper, right? For our friends at home, I would love for you to help me to do this review as well. And then if you want a foolscap, just come on down, show the review that you have put in over there, and you can get the foolscap as well, right? Okay. What thing? Is it wrong? Oh man. Wait, you can just go to like maps. Can you use Google Maps? Then you just search Overmugged Tampines. It works as well. Ren, if I understand it, the answers are going to be like on the soft copy online or something. Okay, so yeah. Megan, hi. I just saw your message, so here you go. Megan, have you got it? Megan, yes, awesome. Welcome. Okay. Yeah, for those at home, I would really appreciate it if you can just do that as well very quickly. Just like show me that you're done. Okay, full scan, done. Awesome, 11 points for you, like wow. And full scan that. Oh, what about part for you? I should go 11 there. You lose. You finish it?
I don't know. Oh, screen time screen over here.
You, that's you. Boys, let's go. What a waste. Hang on. Okay, so as you are filling that in, just a quick look for this one. For next year's, which actually involves IRE as well, you just have to sure, yeah, go ahead. Yeah, can. Okay, so this one is for next year one. So you have a quick look here. Registering the registering the interest early doesn't mean you have to start right immediately. Okay, so can I just have your listen up for a little bit, listen for a little bit here. We actually start our 2026 in like mid-November, but you don't have listen first. Our lessons start typically in mid-November, but I understand that some of you actually like go holiday and stuff, right? Absolutely fine. If that happens, you just have to indicate that you want to register your slot so it doesn't run out, okay? And then I'll see you all next year in January or whichever. Can? Right? But obviously the sooner you can begin with me, the earlier the head start I can get you in our journey towards our O's. Can? Right? Because for example, like with my Sec force now, we're kind of in a bit of a crunch. So the earlier you can start with me, the earlier I can fill in the gaps for all of us, which we have uncovered quite a bit today, isn't it? Okay, so you know what you're working with. The earlier you can start learning with me, the earlier we can fill in the gaps. Okay, so if you want to, the QR code is over here. I'm here. I am either at Tampines or Kovan, right? So you just scan this in there. You can just look at all the details there. Just check in with there. I mean, you need to do this as well. Okay, unless you tell me you don't see me. Okay, so yeah, if you're wondering, bro, Iris is in my class. Okay, all right. Sorry, we don't have one.
Tampines, who want to sit at Tampines?
Well,
Singapore.
I stay in Tampines. That's why I stay here. That's why. That's why mostly. I say not mostly. I'm mostly in Kovan actually. So if Kovan works for you, it's kind of more middle. But green line all the way lah. It's like, right? Okay. Yeah, I will appreciate you making the journey down, right? So this one is just okay. This one actually don't need to stay here to see because it's all available on the website. This one more important. Can? Okay. Friends at home as well. Important information for you. But if not, thank you for the additional 8 minutes. I hope I have helped you uncover some of the gaps. Thank you for sitting through the topics that you are not tested for your end of year still. I hope that it was good learning. And all the best for your end of years. I will see you all soon. Okay? Enjoy the rest of your September holidays. Bye-bye. Bye-bye. Bye, people online as well. Bye-bye. Bye. Yes, awesome. Here you go.
Wait, sorry, how would
Hey,
you
Luana,
set
hi,
the
hey.
answer key?